Hello, everybody. First, I want to thank Arvid for giving me more credit than I deserve. This workshop, I sent a few emails to make this workshop happen, admittedly, but we wouldn't be here without the support of the Institute, and they have done so much more than I have to make this all happen. I also want to especially thank Arvid again for directing credit for this my way. I have a good feeling about this workshop, and I think a lot of us have a good feeling about this workshop. When I sent emails out, I would get responses saying, "Can't wait." It was very encouraging to hear that, but I share it. I think this is going to be a good workshop because all of you have made the effort to come here. I know some of you have traveled a very long distance, and it's much appreciated. And I'm grateful in advance for the stuff you'll bring to this workshop. My job in the next half hour, I want to give us a survey of internal conflicts to talk about what we're talking about when we're talking about internal conflicts. And the temptation was to just do a survey of all my favorite selfish genetic elements, all the weird things they do. Talk about the devil facial tumor, talk about the PSR chromosome, and I have reined that in. I have not fallen victim to that temptation too much. I'll tell a few stories. I'll tell stories later too. But I thought what I would do in this survey is give us some orientation to internal conflicts and try to help us organize how we put it all together. So I'm going to begin here at the middle of the title with the internal piece. The idea that there are internal conflicts is maybe at first surprising. How do we get conflicts inside of ourselves, inside of organisms? Here's 18 cells, 18 protists, and we can imagine that they're in conflict with one another. They're competing for resources, competing for space. If these are mice, they're competing for nesting sites. But what can happen in these evolutionary transitions in individuality is that cells can lump together, things can collect into new kinds of individuals. So that we stop talking about the individuality of those white circles. We start treating this as though there are 4 individuals here, and they may be in conflict. The internal conflict that we then study is sort of like a vestige of what had been those previous 18 individuals. Those parts still retain some capacity to be in conflict with one another. And that's where internal conflicts come from. This presupposes, the way I drew this, I just drew with PowerPoint, I drew 4 red circles and said those are 4 individuals. And it's nice and neat in PowerPoint, but individuality is maybe not so neat in reality. Philosophers have identified 2 styles of ways of identifying what makes an individual an individual. And these 2 different styles break out into evolutionary and organismal types. So if you're thinking about using an evolutionary concept of the individual, looking for criteria to define what makes this one individual as opposed to 2, where do the boundaries exist? What you're concerned with there is fitness. So those red circles have a fitness of their own. We may stop thinking about the fitness of the parts. Once we have a fitness of that red circle, we might even attribute agency and goals to that sort of individual. It may have a goal of maximizing its fitness, perhaps. Some of us talk about the organism as though it's a fitness maximizing agent. We'll hear more from Hannah, I think, after lunch about this idea of maximization in her tutorial. It's an appealing idea. It's one a lot of us use, not without controversy, of course. On the other hand, there's a more organismal notion of what makes an individual an individual. And then that one stresses function more than fitness. If you think about yourself and all of your microbes, or if you want to think about corals and all their microbes, some people will treat the coral plus its symbionts as one individual, because if you were to remove some of those symbionts, it's just not the same thing. So you remove those parts. Those are parts of that overall individual. The function changes when you remove some of those parts. If you remove some of your microbes, maybe you can't digest ice cream after dinner. You're not the same. So again, the focus there is not so much on fitness as it is on function. What is that organism doing? And can it function the same way? That's what makes those white circles parts of that individual, as opposed to individuals in their own right. And I'll say, I think I tend to think more about the evolutionary version of individuality myself, and maybe that's a more popular strain in this room. But the organismal strain, I know we have some people here interested in holobionts, corals, microbiomes, and that's where I think the organismal notion of individuality comes into play as well. And there may be internal conflicts under that notion of individuality, maybe the same, different from the evolutionary notion. I'm envious. Arvid just talked about this. When we proposed this workshop, we use this device of saying, the people who study cooperation and altruism, between organisms. They've got it made. They've got such this nice theory. It's widely adopted. It makes sense. It applies broadly. Yeah, there's an envy about the maturity of the way we talk about external conflicts. The opposite of cooperation. The way we talk about internal conflicts, I feel like we have population genetic models, quantitative genetic models, evolutionary game theory models, ecological models of things like cancer. We'll hear about those tomorrow as well. We have multi-level selection models. We have a proliferation of ad hoc models, ways of modeling these internal conflicts. And a desire of this workshop and a desire that we share is it'd be nice to have a sort of a more mature stance on how we model internal conflicts, how we think about internal conflicts. The multi-level selection one, I'll point out, it maybe is in the running for a way of getting your arms around an entire field of internal conflicts. The social evolution way of doing it too is also another, I think an approach maybe that gets its arms around quite a bit of the field too. And in tutorials we'll hear this morning after the coffee break, Maria Uribe and Jeff Wild will talk about respectively the population genetic multi-level selection approach to things and the inclusive fitness approach to things, which are in the running for ways of thinking about internal conflicts generally. The multi-level selection one, it gives you this between and within individual selection. So my red circles are my individuals, the white circles are the parts within, but those white parts, those white circles may vary. They can experience selection. The red circles may vary one to the next. They may experience selection too. And we have ways of modeling, which I won't show you because Maria will instead, ways of modeling this between and within individual selection. I would just say, I think very often this has become definitional of what internal conflicts are. And hot take, perhaps, or maybe not so hot take, but it's my take on it is that it doesn't capture everything necessarily. It captures a lot, admittedly, but I think it misses some things, which I'll talk about toward the end of this. So here's my survey of conflicts. And again, I'm not going to march through every selfish genetic element, though the temptation is there because they're so much fun. I'll march through a few, but instead I'll give you taxonomies, ways of organizing all the dazzling diversity of internal conflicts and selfish elements. And the three taxonomies I'll introduce come from Burton Chivers, the 2006 book. Another one I'll introduce, but I won't say much about it because we have Andy and Francisco here to talk about it tomorrow. They have a way of organizing internal conflicts. And then with Martijn and Arvid, we've got a way of organizing internal conflicts too. Basically taking all these fun selfish genetic elements, selfish cells and the like, and putting them into different buckets. That's what I mean by taxonomy. So I'll start with the Burton Chivers one because historically it's first. And I think it does a pretty nice job of giving us an order to all this diversity. They have, it's sort of two buckets, but one's doing a lot of the work. The first bucket's quite big. They put almost all of their entire book into the drive bucket. They call it drive, and they're careful not to, the term drive has been muddied. They understand they're using it in acute fashion. They point out three ways by which you might get something like drive-like phenomena: interference, overreplication, and gonotaxis. Now I'll talk about each of those in turn. And then that's basically the entire book. It's chapters 2 through 12, but not chapter 4. Chapter 4 is the one on imprinting, and that one's in its own bucket. And as someone who started in this area by thinking about imprinting, I feel like it's the best bucket. It gets short shrift. But I'll say more about that toward the end here too. But they're a tripartite taxonomy of drive. The interference is the first style of drive they introduce. And I've shown this here with a cartoon of a Drosophila. I'm pretty happy with that drawing. And he's heterozygous, big A, little a. And ordinarily, if meiosis were fair and everything was standard, you'd get an equal proportion of big A and little a bearing sperm. So big A would be one out of every two sperm. But what big A does in this case is, and Rob will talk about this, there are various ways. Big A might contain many genes. There might be a lot of nucleotides in big A, as it turns out. But what it does, the effect it has, is it eliminates little a bearing sperm. So that you basically change the denominator. You can think of the interference mechanism as one that changes the denominator. It's pushing down on everyone's shoulders to look taller. So now the ejaculate contains nothing but big A sperm. So it's killing off the competition. That's maybe the key feature of interference mechanism, is that it kills the competition. There's real like knives out here. in this kind of interference. And again, this one fits neatly with this multi-level selection approach because within males, there are big A's and little a's, and the big A's fare better. They have higher fitness. But between males, some of those males, in that case, the way I drew them, they might be only half as fertile as other males. That's not always the case. Palm, palm stalk-eyed flies have totally normal fertility despite having this sort of drive. But ordinarily, we think that males that have these driving elements have lower fitness. So selection between males favors the non-lethal version of the allele of the gene. Overreplication is their second kind of driver. Again, driver in scare quotes. Here I've drawn one transposable element, and it's one of 20,000 genes in the genome, just for a rough number, 20,000. And what this transposable element does, it's a DNA transposon. It cuts itself out of the DNA and then reasserts itself somewhere else, but it cuts itself out of the DNA after the replication fork has passed through it, and then inserts itself somewhere before the replication fork has gotten to it. And so it gets replicated twice during DNA replication. And you end up getting, and I realize my math is wrong here, because it changes the numerator. That's the important thing. I guess it changes the denominator too. For any pedants that want to say that should be 20,001, I hear you. Yeah. But the point is it's making more copies of itself. That's what these overreplication elements do, is they just make more copies of themselves. And that's just one variety of transposable element. There are loads more. Cancer can be thought of as in the same fashion. It's overreplicating. So here I am going for a walk this afternoon. I'm out in the sun. The sun's blasting down on me. A cell on my forehead gets blasted with a solar ray. And now it's, instead of having one cell, it gets mutation, starts proliferating. And again, there should be 36 trillion and one, but it changes the numerator as opposed to the denominator. So there's no killing off in these overreplication mechanisms. They're just adding more. So not as mean and nasty, perhaps, as the interference type. Nevertheless, I feel like there are, I think there may be more elaborate, better studied defense mechanisms against these kind of overreplication types, possibly because they're just so numerous. There's so many ways of transposing, so many ways of overreplicating as a cell. And so we'll hear more about maybe some of the defense and some of the tissue design, some of the organismal design in light of the fact that we are prone to cancer in Steve Frank's tutorial a little later on. We might hear more from Justin, too, about silencing or repression. Actually, I'm sure we will. Justin can't stop talking about silencing or repression. He will talk about that in his talk tomorrow. The third kind of Burton-Trivers driver is gonotaxis. And this one is drive. So for any pedants that were correcting my math before, you'll like this. Meiotic drive is what I'm showing you here. This is drive that happens during meiosis, not after, not before. So originally the term referred to this kind of drive. We have a female monkey, or we have a monkey flower here, and in female meiosis, she's heterozygous, big A, little a. And what the big A allele does is orient itself on a spindle such that it makes its way to the egg pole. And that first division in meiosis, there's one winning side and one loser side. And the name of the game is getting yourself to the winning side. That's what gonotaxis is. There are a variety of cell divisions during multicellular development where you may go germline or soma. And if you can get yourself into the germline, that's gonotaxis. If you can get yourself with a chromosome into the egg pole as opposed to the polar body, again, that's winning. And that's selectively favored within a body. It's just a neat little trick that these often centromeres play during female meiosis. And what's nice about these, in stark contrast to the interference types, there's no murder here. They're just riding along. The spindle was already going to the egg pole, and it's just a matter of getting yourself onto that track. So stowaway, I think of them as stowaways. This is maybe the dream of all selfish elements, is to cause very little ripple at the surface, to cause very little organismal level fitness consequence. And some selfish elements can pull this off. They can be just the perfect kind of stowaway where you hardly notice they're there. And indeed, and it's not just the gonotaxis ones that do this. There can be some amelioration of the harmful effects of transposable elements and the like, so that there's less organismal defense against these selfish elements. They'll still spread despite being selfish and no good to the organism whatsoever. So that's the Burton-Trivers division, three-part division of drivers. There's a few that don't quite fit neatly into that taxonomy. One of them is homing into nucleases. It's a little bit of overreplication and a little bit of interference. The way these work is we have a heterozygous individual here. The homing endonuclease gene is in that box. It encodes a nuclease that targets about a 30 base pair recognition sequence. So it's there in the wild type sequence. It cuts it, and that cut, double strand break, looks for homology to repair. The neat trick of the homing endonuclease gene is that the 30 bases are split on either side of that HEG box. And so the homology, the broken allele, finds homology in the HEG and then uses that as a template. So you end up going from heterozygous to homozygous. The HEG just copies itself over. So it kills off the alternative allele, but it makes more copies of itself too. It doesn't quite fit neatly into either overreplication or interference. There are some selfish centromeres, again, during female meiosis, and Lawrence Hurst has written about these recently, and there are some B chromosomes in grasshoppers and things that do this too. The trick they employ is they just don't attach to the spindle at all, and they just kind of camp out in the middle of the cell as it divides. And it turns out that first division isn't symmetric. More cytoplasm ends up in the egg pole. And so if you just camp out in the middle, you make it to the egg pole. It's a really, you might have thought these were clever. It's clever in a way, but it's just lazy. But that works, as it turns out. So big A there is going to ride to the egg pole, even if it wasn't being pulled there. If it attached to the spindle, it might have ended up in the polar body. This one ends up in the middle and in the egg, but along with the little A allele. So now this egg is doomed to be aneuploid. That embryo will die early. And that's fine from the big A's perspective. It was going to die anyhow in the polar body, and this way it kills the competition. So it's a little bit of gonotaxis. It's a little bit of interference. It's not quite, nature doesn't disobey the law of nature. Nature disobeys the law of naturalists. I think I'm paraphrasing someone there. This taxonomy isn't totally neat. I just want to show you this drawing I did. I'm quite pleased with it, of Andy and Francisco. They have, and they'll talk about this, they have a division of intragenomic conflict into 3 buckets. And it's not so much about the mechanism by which these things achieve their success. It's more the motivation for why they're in conflict in the first place. That's how it breaks down. So rooted in social evolution theory, inclusive fitness, genes may be in conflict because of where they came from, their origin, where they're going, that's their destination, next generation, or where they sit currently. And the transmission genetics, which I think Jason will talk about as well in his tutorial, the transmission genetics sets up some of these intragenomic conflicts and these internal conflicts. I'll say nothing more about it, but I'll just pause while you have a look at that. It's some of my bit. It's Rembrandt. Yeah, not to scale. Yeah. All right. The final taxonomy I'll introduce is one I'm quite fond of. This is from myself and Martijn and Arvid. We see self elements being broken into 2 buckets. There's a transmission distortion bucket and a trait distortion bucket, and everything from that drive bucket in Burton Trivers fits into the transmission distortion bucket. It's all about part versus part, making more of yourself within the organism. And it lends itself to this multi-level selection perspective. So I think that multi-level selection perspective explains a lot of biology, can do a nice job with that. It's this trait distortion bucket where I think it falls down. I think the reason it does is that the kinds of conflicts we're dealing with there are of a different nature. It's not so much that it's part versus part. They're not competing within the organism or within the individual. They are in conflict with the individual instead. So there are traits that would benefit the part, but maybe harm the organism writ large, and vice versa. They're in conflict over what the trait of the organism ought to be. I'll give you an example of this. It's not as good a drawing, admittedly. Here's a pregnant mouse. Here's a mouse pup, a mouse embryo with a placenta. And that Z there, that's the trait value of this placenta. The question is, what trait value should it take? Should it be a big placenta, small placenta? Should it be demanding, not so demanding? Under parent-offspring conflict, which we'll hear about tomorrow from Abdel, and maybe Ralph too, under parent-offspring conflict, we appreciate and have for half a century that mothers and offspring don't agree on what that Z ought to be. They have different inclusive fitness optima for that trait. But it turns out, since David Haig's work in the last few decades, we can also think about the genes within the embryo itself. It turns out they don't agree necessarily on what the optimal placenta ought to be, how big it should be. Different values of Z might maximize the inclusive fitness of maternally derived genes versus paternally derived genes. So there's an internal conflict here over how big a placenta you should make, how demanding you should be on your mother. It's not one of these drive-like phenomena. The alleles within that mouse don't have different fitness within the mouse. They may have different inclusive fitnesses, but has a different flavor, different structure to the part versus part transmission distortion types of conflicts. Another possibility is that here's a plant, and the flowers can be male, female, hermaphrodite. You can think of the trait Z being how much they allocate to male or female function. And again, they don't agree. The parts within the plant don't agree on what the plant ought to do. So mitochondria, which are transmitted maternally, think these should all be female flowers, think so to speak. But they have maximal fitness if these were female flowers rather than allocating anything to male function. Other genes in the genome, the plant overall, may have a different optimal Z in mind, so to speak. I think my perspective on these trait distorting conflicts are these are the real challenging ones because these are traits of the organism. We often think that organisms wield a phenotype to maximize their fitness. And again, if we're comfortable with the maximization sort of language, but again, it's the organism wielding that, but there could be parts of the organism that wield that same phenotype. And so parts and organisms can be in conflict over what phenotype to wield, so to speak. So I find these theoretically, these are the most exciting and tantalizing kinds of conflicts, possibly showing my bias coming from having studied imprinting first. All right, I'll end there. I hope I've given you a way of organizing internal conflicts. I have a few minutes for questions, either about this stuff or the workshop overall, and then we have a break. So yeah, I'll take questions.
Yeah, no, that's a really great summary. So I was just wondering with your two buckets, so what about something that does transmission distortion, like a driver that also changes the trait?
Yeah, good question. This is where it gets tricky. The way I think about it, let's see if I can untrick it. If the question is what is the optimal sex for this offspring to be, or what sex this body should be, I don't think an X chromosome per se benefits from the body being female, right? So there are driving X's that cause their descendants to be female. So if an X drives against a Y, it's going to be in a daughter. But being a daughter actually harms that X chromosome's fitness, if anything, because now it's assigned itself to the more common sex. So if we ask what trait value would that driving X chromosome prefer, it would prefer a male offspring, if anything, or it would prefer a 50-50 offspring. It might be stuck with the usual stable strategy of 50-50. But nonetheless, I think the way that driving X chromosomes succeed is by transmission distortion. They just have this as a constraint. They also cause this trait value to change. But I don't think they're changing it in the direction that favors them. So neatly, if I'm thinking about why are these two buckets, what transmission distortion is about is simply about just like changing the numerator denominator, making the numbers work. Trait distortion is asking what value for this organism is optimal, and do the parts have a different optimum in mind from the collective, from the organism? This has been Tom and I have talked about this too. It's one of those. This is the one element, the one kind of the one selfish element where it's like, is it trait distortion or transmission distortion? I think I can neatly tease apart those two kinds of distortion. Sometimes there are elements that do a little bit of both. But I think of the driving X more as a transmission distorter that happens to also then do something funny with sex ratios. But it's not doing it as part of its strategy. Its strategy is to just transmission distort. Cytoplasmic male sterility, its strategy is to change sexes, or change the sex of the bearer, if that makes sense. It's again, I'm special pleading to atomize the effects of these drivers. But
so along those lines, I realize Wolbachia is a weird element because it's a different organism, but so is the mitochondria. That Wolbachia, especially when it's killing males, is killing males presumably to benefit those females. So where does that fit?
It directly doesn't benefit, right? It indirectly benefits by helping sisters, let's say. Right. It's a trait distorter. It's not that, but yeah, I suppose the thing you're focusing on there is a litter, let's say, or a brood.
Yeah.
What's the optimal sex ratio in the brood? From the perspective of the Wolbachia, all female might be the best sex ratio. From the perspective of a nuclear gene, something different. So yeah, I'm thinking again, a trait distorter, not a transmission disorder. It's achieving that by changing the organismal phenotype.
Maybe just to add on here is that, of course, ultimately it's all about the spread of different alleles in a sense. And so in the end, it's, what transmission you acquire, but it's really if that the route you take to success is through interfering with the rules of transmission as dictated by ontogeny or whatever, or by reshaping that ontogeny to do something to your bidding, kind of indirectly getting there. So that's of course for genomic imprinting, it's not about necessarily changing the rules of transmission, but you actually build a more demanding organism, which will accrue higher, resources and thereby produce more offspring in the next generation. So it's the root, it's really about like, what is your way to success? But of course, success in the end is just frequency change, I guess.